Welcome to the third and final installment of modules for Prime Immunology. Our previous two modules have covered both the innate and adaptive immune systems. Throughout this module, we will address key concepts of the complement system. The complement system acts as an enhancer. It complements the functions of antibodies and phagocytes to help clear microbes during an infection. At its core, it serves as a liaison between the innate and adaptive systems. The complement system holds three critical functions. First off, opsonization. Opsonization is a process during which microbes are coated by specific proteins in order to be more easily identified by phagocytic cells for targeting and destruction. Complement-induced inflammation occurs when the system itself induces an acute inflammatory response, resulting in dilated blood vessels and immune cell recruitment to the site of infection. Lastly, lysis occurs when a complex called the membrane attack complex, or MAC, is formed by complement proteins to come in and penetrate a pathogen's outer cell wall, to then break up or lyse the agent. The complement system is made up of over 30 proteins and factors, most of which are synthesized in the liver. Serum proteins are named with the letter C, followed by a number, and their fragments contain an additional letter, usually A or B, to denote that the protein has been spliced. You will become very familiar with the many components of complement, as well as their nomenclature, during your immunology block. As a system, there are three pathways of complement activation: the alternative, lectin, and classical pathways, in this order. Though activated separately, the three pathways end on one common step: cleavage of the complement C3 into C3A and C3B. In the next few slides, I will introduce you to the key components of each pathway. Keep in mind that there are more intricacies involved, which you will learn about soon in lecture. Despite its name, the alternative pathway is the first one to be activated. C3 is a main complement protein that is continually produced by the liver and released into circulation. As a small amount of C3 is cleaved to form C3A and C3B, the resulting C3B fragment can either bind a microbe if infection is present or be inactivated during healthy state. If C3b does bind a microbe, it signals the formation of an enzyme called C3 convertase through a series of factor-mediated steps. C3 convertase acts to produce even more C3b fragments, and thus a positive feedback loop. Hence, activation of the alternative pathway. C3b that remains bound to the pathogen surface ultimately acts either as an opsonin or it undergoes further steps for the eventual formation of an enzyme called C5 convertase, which forms the membrane attack complex. Sequentially, the lectin pathway is the next one to be activated in complement. A protein known as mannose binding protein, MBP, exists normally in circulation. If MBP binds to mannose on a bacterial surface, it becomes activated to produce an enzyme that cleaves the complement protein C4 into fragments C4A and C4B, as well as C2 into C2A and C2B. Fragments C4B and C2B join to form a C3 convertase, just as in the alternative pathway, which, as you remember, cleaves C3 to generate C3b. Ironically, the classical pathway is the third and final of the three complement pathways, and it is unique in that its activation requires antibodies. Remember, it takes time for antibodies to form, therefore it makes sense that this would be the slowest pathway to be initiated. Also remember, IgM and IgG are the two types of antibodies that can activate complement. When antibodies bind bacteria, the complement protein C1, which is present in its inactive form in circulation, also binds to the antibody at another site. This induces a conformational change in C1, leading to the production of an enzyme that, similarly to the lectin pathway, cleaves C2 and C4 into C2A, C2B, C4A, and C4B. Again, fragments C4b and C2b join to form C3 convertase, which, as you remember, cleaves C3 to generate C3b. Provided is a brief summary slide on the basics of complement. Thank you.